Welcome to BIOL 100. This will actually be both Chapter 2 and Chapter 3. Alright, so this unit, or this lecture is over quite a bit of information. So your text actually goes much more into depth on a lot of it. So don't get too caught up in all of the little details, but instead focus on the main concepts that are kind of covered here, and then go through the text and make sure that you understand these concepts. But I'm not worried about you remembering the tiniest details about things. Okay. So we're starting off with the smallest thing we can possibly think of, which is an atom, right? All matter is composed of basic structural units. We start with an atom. That's our smallest unit of matter. It can't break down chemically. And then we go to an element, which is a type of substance that is made only of one type of atom and has distinct chemical properties. Then if we join two or more atoms together and they bond chemically, like in the case of H2O, water, then we form a molecule. So just real quick, atoms are made up of protons, neutrons, and electrons. You've probably heard of this or remember it maybe even from high school. Our protons are our positive guys. They're in the nucleus here in the center. Our neutrons are neutral. They're also in the nucleus. And then our electrons are the negative guys that are hanging out on the outside, just wanting to get their negativity in. So we're going to cover this very quickly, but what's the difference between atomic number and atomic mass? So if you've seen a periodic table, has all the table of the elements, atoms of each element do have a standard number of protons and electrons, and that is what will give them their atomic number. So atomic number is the number of protons, and again, each element has a distinct atomic number, while the atomic mass is the actual mass of the atom, which is roughly equal to both the protons and the neutrons. Okay. Number of neutrons can vary in an element, and elements that have different numbers of neutrons are called isotopes. So it's essentially the same element, but it's kind of got a little spin on it, if you will. Can't think of really the best way to describe an isotope other than it's the same element but just a different number of neutrons. So it is slightly different, and again, electrons don't need to be included in the calculation of atomic mass. It's just the neutrons and the protons. Okay. So we can calculate the number of neutrons in an element by subtracting the atomic number, so the number of protons, from the atomic mass. This isn't something we'll do in class. It's just maybe kind of good to know. You see this as you're going through not only the text, but maybe other parts of science in your everyday life, or as you keep going into further classes in college. But again, our atomic mass versus atomic number. So you can see our atomic number here. This is carbon, so if we're looking at the periodic table of elements, and then our mass number is down here on the bottom. So you can see atomic number of 6, and there are 2 stable isotopes with mass numbers of both 12 and 13 respectively. So we have carbon's 12 atomic mass is approximately 12.11. So again, isotopes forms are different forms of an element with different numbers of neutrons, and they also have different mass numbers. So you can see protium, deuterium, and tritium. So this is basically just different isotopes of hydrogen, right? So we have 0 neutrons, 1 neutron, and 2 neutrons. The main reason why I really wanted to talk to you about isotopes, we're not going to go into them very far in depth, but you may have heard of radiometric dating. Sometimes it's referred to as radiometric carbon dating. And I wanted you to kind of understand what an isotope is so that hopefully when you see this come up, you sort of understand the basis of it. And basically what it is that radioisotopes, which are just isotopes that emit neutrons, protons, and electrons, we can use those to radiometrically date whatever it might be. It might be fossils, or if we find something in the ground, right? Again, this type of carbon dating is a little bit newer. We've been doing it for some time, but it's really well refined now. So you can see an example of this is over time, carbon-14 actually decays into nitrogen-14. And we know the time of that, and we know half-time, things like that. So we can compare the carbon-14 in the atmosphere to the carbon-14 in fossils remaining, and then that way we can actually estimate the fossil age. So there is quite a bit of guessing here, and there is, a little bit of room for error as you're talking about billions or even sometimes lesser and millions of years. But overall, we can come up with a pretty good guess. And for older objects, they can analyze the presence of isotopes of, say, uranium, which decays to lead. You could also use potassium or rubidium. So this is really common when you're looking at fossils as well. Okay, so kind of switching gears, and now we're going to go into the molecular side. When we join these atoms together and they form molecules, we do this through what's called covalent bonds. So this is just when atoms share a pair of electrons, and if we want to make them stable, what happens is the atoms actually fill their orbit. So remember, our negative electron is orbiting out here. So you can see the different orbits. We have the first, the second. The first just has 2 electrons. The second and above have up to 8 electrons. So having said that, one of the main elements that we'll really focus on in this class and that you'll hear quite often in biology is carbon. And carbon is essential for life. We went over the properties of life in the last lecture, but one of the other properties of life as of right now is that it's carbon based, because everything that we know that is living on Earth is carbon based. And carbon is essential. It also has up to 4 covalent bonds, so it's a very stable molecule. Okay, so it's got 4 electrons in its second shell, and it can make 4 covalent bonds with other molecules to total 8 electrons. So again, a very stable molecule, and it's one of only 6 elements that make up most of humans' weight. So you can see oxygen's our biggest one at 65%, but carbon is second at about 18%. Really, there's just a handful of elements that make up not even just humans, but we can see the atmosphere, right? We've got about 21% oxygen in the atmosphere. The other 78% is nitrogen, and then we've got about 1% that's kind of just trace elements. So we do have a little bit of carbon, a little bit of hydrogen. You can see kind of similar for the Earth's crust. Oxygen is still our major player here. And then for humans, we are carbon based. We are made up of about 18%, and then a lot of oxygen, and then a few other things in there as well. So again, with carbon being a very versatile component, we then can refer to anything that has a carbon-based backbone, which means that it has at least one carbon-hydrogen bond. You can see these little CH bonds here. We refer to those as organic molecules. So don't get this confused. Obviously, you've gone to the grocery store and seen organic versus not organic produce, let's say. That's a little bit different. Then we're talking about pesticides, right? But here we're talking about organic in terms of life. This would be biotic, is organic. And so since we consider life to be carbon-based, in order for it to be an organic molecule, it's got to have a carbon-based backbone. On the contrary, inorganic molecules lack carbon-based backbones and therefore do not have these carbon-hydrogen bonds either. So talking about the molecules of life, we have some macromolecules, macro meaning big, right? So these are our large organic molecules, and we've got 4 different ones. So all of them are composed of subunits called monomers, mono meaning one. And then our monomers can link together and form poly meaning multiple. They can form polymer chains. So our 4 molecules, we've got four different ones that, again, make up all living things. We have carbohydrates, proteins, lipids, nucleic acids. You'll notice we'll get into this a little bit more in the nutrition chapter closer to the end of the book when we start talking about digestion as well. But you'll notice this is essentially what we take in our meals as well, right? Carbohydrates, proteins, lipids are basically the same as fats. Nucleic acids, we don't, but these are what essentially go hand in hand with our nutrition. And all of our chemical reactions that do involve those macromolecules, the carbohydrates, the proteins, the lipids, and the nucleic acids, all of them are catalyzed by enzymes. And catalyze just means that our reaction rate is going quicker, or we're speeding up that little time there. And enzymes are just a biological molecule that can speed up the reaction. So they'll speed it up either in hydrolysis or dehydration reactions. And you can see dehydration, we are forming new bonds, which requires energy, or hydrolysis, we're breaking bonds and releasing energy. Not so concerned with you knowing this part, just really want you to know what an enzyme is and that it catalyzes reactions or makes them a little bit quicker and make sure that those reactions go. And you'll see specific enzymes that exist for each different kind of macromolecule class. Our carbohydrates are broken down by enzymes like amylase, sucrase, lactase, maltase. You'll see that we always end in A's for our enzymes. Our lipids are broken down by lipases. Proteins are broken down by pepsin and peptidase. So again, these are catalysts and biochemical reactions, and each enzyme has a specific substrate. So this is kind of like a lock and key mechanism. If there's not an enzyme that can go to, like, let's say, a specific protein, then it's not going to happen. It can't just catalyze any reaction. It does have to be a lock and key where it actually fits in. And again, there's different types of enzymes. We have catabolic, which is breaking down, anabolic, which is building up, and catalytic, which can affect the rate of reaction. The way I remember this is that I'm allergic to cats, so cats kind of break me down. I don't really have any other interesting mnemonic for this, but catabolic, breaking down, anabolic, building up. Okay, so let's go into a little bit more about these macromolecules. First up, we've got carbohydrates, and I'm sure you know a little bit about carbohydrates based on how you eat normally. But carbohydrates are made up of sugar monomers. Okay, and then we can form more complex carbohydrates by joining these together. And carbohydrates are often found in grains, fruits, vegetables, and they provide energy to our bodies in the form of glucose, right? Represented by the general formula, carbon, water, and then to some extent, right? So carbon, hydrogen, oxygen is 1, 2, 1. And we've got a few different subtypes. We have monosaccharides, again, mono meaning 1, disaccharides, 2, and polysaccharides, 3. These are essentially different kinds of sugars. Okay? Monosaccharides, not too concerned with you knowing all of these little things, but just know that they have about 3 to 7 carbons, and these guys end with the suffix "ose." So aldoses, ketoses, trioses, pentoses, hexoses. All of these are different carbonyl groups in the carbon chain. You can see we name them based on how many carbons are here. But if you see an ose, the likelihood is that that's some examples of monosaccharides, like ribose, glucose is the most common one. That's basically our energy formation, right? We're breaking down our food into glucose essentially to convert it into ATP and things like that, which we will talk about later in the semester. We also have galactose and fructose. I'm sure you've heard about both of these. Again, glucose is probably our most important source of energy. Galactose is part of lactose, which is a milk sugar, and fructose is part of the fruit. So those are some of our common monosaccharides. We also have disaccharides. So this is when two monosaccharides link up. So we might get glucose and fructose that come together and they will create sucrose, which is a disaccharide. These are joined by a glycosidic bond and they also release water. Again, not too concerned with this part. Just know that things like sucrose are disaccharides and that's just combining our monosaccharides. Other common disaccharides that you might see, maltose, which is a grain sugar, lactose, again, milk, and sucrose, which is our general table sugar that we always refer to. All right, so we have mono and disaccharides. Now we're getting into polysaccharides, which is 3 or more. So this is a long chain of monosaccharides, again, joined by a glycosidic linkage. They might be branched or they might be unbranched and consist of multiple types of monosaccharides. They can be quite large. You can see here molecular weight over 10,000 Daltons is relatively large when we're thinking about molecular weight. And these can form things like cellulose. Cellulose is a polysaccharide that's found in the cell wall of plants. Gives it kind of a fibrous structure and helps provide some structure for the plant as well as kind of an extra barrier of protection. We also have chitin, which is the hard exoskeleton of arthropods. It has this polysaccharide chitin in it that also has a little bit of nitrogen in there. So going into some examples of carbohydrates. Oh, sorry, I skipped ahead a little bit fast, but we have both simple and complex carbs. And you've probably heard about this if you've thought about nutrition or taken nutrition classes or anything like that. But our simple carbs are the ones that we kind of want to avoid, right? Both of these are made by green plants, and both are broken down into glucose for energy. But our simple carbs are things that have very little nutritional value, like soft drinks, sugar, milk, fruit, and our complex carbs actually take longer to break down, which means that usually it can help you feel full a little bit longer. And more importantly, it essentially takes longer to break down and actually go through the whole reaction. And that kind of helps stabilize like your blood sugar and things like that as well. So examples of this would be corn or peas, whole wheat bread, vegetables, oat bran. Again, take longer to break down. Most of them contain fiber as well, which helps keep our digestive system healthy and make you feel full for a longer period of time. Okay, up next we've got proteins. These are our most abundant organic molecules and they have a diverse range of functions. So we're not just talking about proteins that we're consuming here. We're talking about proteins in terms of different proteins in your body that have different functions. Again, they might be regulatory, structural, they could be protective, they could be used for transport, they could be enzymes, again, catalyzing reactions, or they could be toxins. Proteins are made up of amino acids, which are just monomers that make up a protein. Um, Again, we've got kind of a fundamental structure here. We've got our carbon-based backbone, right? We have at least one carbon-hydrogen bond. We have a carboxyl group over here, and then a hydrogen group, and we've got another side chain or an R. Each amino acid has a different R group, that variant that I was just showing you, that side chain. And we've got 20 different common amino acids commonly found in proteins. The R group is what determines the chemical nature of each amino acid. So they could be nonpolar, they could be polar, or positively or negatively charged, or they could be nonpolar aromatic. Amino acids are represented by a single uppercase letter or 3 letters. So, for example, valine could be V or it could be abbreviated VAL, aspartic acid, D, or ASP. You might see it both ways. Our body also needs what's known as essential amino acids. So these have to be found in your diet for humans. We do not manufacture these as part of all of our different regulatory processes in the body. So we must take this in through our diet, through our nutrition. Okay, so things like isoleucine, leucine, and cysteine, those are all what are known as essential amino acids. So, why are we talking about amino acids? Well, amino acids are what make up a protein. So the sequence and number of amino acids that we have is what actually determines our protein shape, size, and function. The shape of proteins is crucial to their function. So if we have one that's misshapen, if it doesn't synthesize correctly, in most cases that means that the protein is not going to do anything really. And protein shape is based upon 4 levels of structure. We're not going to get too far into this, but just know that there's primary, secondary, tertiary, and quaternary structure. So we can see the primary structure here is just the unique sequence of amino acids in a polypeptide. So we can see that it's pretty much linear at this point, right? And that amino acid sequence is based upon gene encoding that protein. So we have different genes within our genetics that say what proteins our body should encode, and any change within the nucleotide sequence of that DNA could lead to a change in the amino acid. If that happens, it would then lead to a change in protein structure and function. So here's an example. If you've ever heard of sickle cell anemia is something that we don't want, right? It means that we have sickle cells instead of normal hemoglobin, which means that we've got good oxygen in the blood, and we're not anemic, essentially, where we're not getting enough oxygen into our blood cells. So you can see here, this is our primary structure. So you can see our linear protein here, the different amino acids that we have, and we have one amino acid that is changed. Okay, and because of that, instead of having our normal beta subunit in our secondary and tertiary protein structures, now we have a sickle cell, so kind of misshapen, and because of that, we get the capacity to carry oxygen being greatly reduced compared to what it would be if our proteins were functioning normally. So the result of this one single amino acid change out of 600 amino acids that code for human hemoglobin can now cause these crescent-shaped sickle cells to come to fruition instead of our normal cells here, as we can see in this blood smear that's under the microscope. So to sum it back up, our proteins are made up of these amino acid monomers. They make a linear strand, which is our primary structure. And then after that is when they go and they fold into our tertiary, quaternary, or secondary, tertiary, or quaternary structures as well. That's what we want to make sure goes according to plan, essentially, for our proteins to behave the way that they were meant to. Up next in our molecules of life, we have nucleic acids, and these are made up of nucleotide monomers. So we can see here a nucleotide has a core structure. It has a phosphate group over here, along with a sugar and a base. Okay, so this varies slightly between DNA and RNA, which we'll talk again a little bit more about once we get into genetics, but just to kind of quickly introduce you into what DNA and RNA are. RNA molecules consist of only one linear chain of bonded nucleotides, while DNA is our classic double helix that you've probably heard a little bit about. And it's got 2 chains of bonded nucleotides twisted into that. So our nucleic acids, this is what actually gives us our genetic material, right? And there's 2 different types of nucleic acid. We have deoxyribonucleic acid, which is our DNA, again, our classic double helix, and our ribonucleic acid, which is our RNA. Our RNA is essentially what is, our DNA is what has the codes, the genes that tell us what proteins to make, and our RNA is actually the one that's going to transcribe and translate those. So our nucleic acids are found within the nucleus of eukaryotic cells. They are also in mitochondria, chloroplasts, which are only in plants, and prokaryotic cells. So what is DNA? Again, this is going to be kind of a quick introduction into it just so you know a little bit about it, but we'll get more into it later in the semester. But DNA codes for the genome of a cell. It has all of your genetic content. Everything that says whether your eyes should be blue or you should have a certain disease, all of that is within your DNA. And it's bound up into this stuff called chromatin, which is just a complex of DNA and histone proteins that are holding it all together. And then that will then lead into chromosomes, which are our thread-like structures that are containing tightly wound and packed chromatin. And again, our DNA is coding for thousands of genes, and these have the instructions for producing proteins or various forms of RNA. Our DNA controls all cellular activities by turning our genes on or off. And RNA is pretty much the one that's involved in protein synthesis. Synthesis just meaning basically creation. And we've got a few different types of RNA. We have mRNA, which is our messenger RNA, and it's what goes through what's called transcription. So it's basically got the blueprint. So it sends the blueprint over and says, okay, here's what we're going to make. And then we have a little bridge between our nucleotides and amino acids. That's our tRNA, our transfer RNA. And that is how we are going to translate our blueprint and turn it into an actual amino acid strand, which will then become a protein after it's folded correctly. We also have ribosomal RNA or rRNA, which assists in the protein synthesis or creation of proteins. DNA and RNA both have monomers known as nucleotides, which we kind of talked a little bit about. Has its nitrogenous base, a pentose sugar, meaning 5 sides, and 1 or more phosphate groups. So you'll see for DNA, we have cytosine and thymine, and adenine and guanine. So these always go together. A and T, adenine and thymine, always go together, and cytosine and guanine always go together. However, when we have RNA, we no longer have thymine. So now we have adenine with uracil and cytosine with guanine. Okay, so this is what's referred to as complementary base pairing. And again, we'll get into it more later, but these pairs always go together, and that's what forms the rings on the ladder of our double helix. Okay. So in our sugars that we find here, we have deoxyribose, which is found in DNA, and then just ribose, which is found in RNA. So again, these are our bases right here that are making up the ladder of the DNA double helix. The sugar and phosphate lie on the outside. And this is important too because when we are talking about covalent bonds, we also have something called hydrogen bonds. So each base here connects to the other base on this side between, and in between they have a hydrogen bond with the base from the opposing area. On the outside here, we have our our sugars and our phosphates. Those have they're very stable. They have a very strong backbone and provide a strong backbone for our DNA so that it doesn't just unravel at any time. And when we put our base pairs together, they run anti-parallel to each other. So one runs from 5' to 3', while the other runs from 3' to 5'. And again, adenine will form hydrogen bonds or base pairs with thymine, and guanine will pair with cytosine. So you can see these little hydrogen bonds right here, grouping these two bases together. And you'll see that they're dashed lines, and that's because they are very weak. So they're able to break this very easily so that our double helix can unwind or unzip itself. And that's when we can go through the process of replicating our DNA. Okay, so again, our DNA can express a particular gene by synthesizing RNA via the process of transcription. So transcription comes first, and then later will come translation. Once we have an RNA-based sequence that's complementary to our DNA sequence, we have RNA, our base is uracil, is in place of thymine. So again, once we transcribe it, let's say this is our DNA sequence AATTGCGC. Our mRNA complement would be A goes with T normally in DNA, but with RNA it'll go with U. And then we have another A, so we have a U. We have a T, which will go with A, G goes with C, and then C goes with G, G, C, C, G. So that's what we mean by complementary base pairing. We're basically just pairing it with the opposite one that it goes with. Okay, now the production of proteins. So we have ribosomes on some of our organelles within the cell, and these are what make up proteins as well as our RNA. Remember that ribosomal RNA. So our mRNA, our messenger, binds with the ribosomes, and then the rRNA has catalytic activity, so it's going a little bit faster, and the bases of the mRNA are read in sets of 3 bases. The 3 bases together we refer to as a codon. And then our transfer RNA is going to pair these codons with the correct amino acid. So say we have 3 codons like A, T, G. That will go to the right amino acid, and then that will link up the amino acid in place there. And these peptide linkages are made at the ribosome as the polypeptide continues to grow. So we basically have all of these RNA letters, right? And as we start getting them, we put 3 together, and now we have a amino acid. And then we start linearly collecting these amino acids that will then form the protein. Okay? So a ribosome has 2 parts. It has a large subunit and a small subunit. And the mRNA sits right in between these two. Okay, so you can see it here, the small unit as well as the large unit. mRNA is sitting right in between them, and a tRNA molecule is going to recognize a codon on the mRNA, bind it to a complementary base pair. So we have our growing peptide chain here. And then it's going to add the correct amino acid to this chain. Okay, I know that was a really quick explanation of DNA, and again, we'll get more into it, but I just want you to kind of have some of the basics because you will see a little bit on it, not just in this lecture, but in a few of the lectures to come as well. Okay, the very last of our 4 macromolecules that we are talking about now is lipids. Lipids are a really diverse group of nonpolar hydrocarbons. Hydrocarbons are actually hydrophobic, means that they're water-fearing. Phobia, phobic, as in fear. So you can see an example here on this little otter. This has hydrophobic lipids within the fur that kind of protects these mammals from the elements. So it's almost kind of like a waxy coating, if you will. And lipids are made out of repeating monomers. So you'll see here we have kind of the same thing going over, and good examples of this are we have saturated and unsaturated fats, we have steroids or sterols, as well as different fatty acids. Functions of lipids, these guys are for our long-term energy stores. So when we start talking about nutrition and digestive enzymes and things like that, the lipids are actually our fats, right? Our long-term energy stores. They provide insulation from the environment. They serve as building blocks for some different hormones. And they're an important component of cellular membranes as well, which we'll talk about here in a second. There's a few different types of lipids. We have fats, oils, waxes, phospholipids, which is our cellular membrane, as well as steroids. So our fats contain 2 main components. They have glycerol as well as fatty acids. And we can see here within the fatty acids, we have both saturated and unsaturated. So means it contains no carbon-carbon double bonds in the carbon backbone if it's a saturated fatty acid. These ones are packed tightly and they are solid at room temperature. So saturated fatty acids are things like butter or fat in meats that you see that will melt down pretty fast, but they're actually solid at room temperature. And these are ones that we kind of want to stay away from, right? We want to limit these things in our diet because they could potentially lead over or be associated with cardiovascular disease. Now we have our unsaturated fatty acids. So this one has at least one carbon-carbon double bond within our carbon chain backbone. So we also have monounsaturated fat and polyunsaturated fat. Mono meaning one double bond and poly meaning more than one double bond. Most unsaturated fats are liquid at room temperature, so this is things like oils, generally preferred to our saturated fats, but still want to kind of watch the amount we put in our diet of these. So we've also heard of trans fat, right? And basically what it is that each double bond within that saturated fat, they can be in one of two positions. So they could be either cis or trans. Cis means that our hydrogens are on the same side. Trans means that the hydrogens are on the opposite side. And our cis acids basically will just get a kink in the chain. So they can't be packed too tightly. These are the ones that are liquid at room temperature, our unsaturated fatty acids like our oils. And then we also have trans acids. So this one has no kink and we can create this through processing. So these are, again, the things that we want to avoid too much of in our diet because these are things that can increase our LDL cholesterol, which is the one that's bad for you. We have two different kinds, right? HDL and LDL. LDL is the one we want to stay away from. That's pretty bad for our heart. We also have essential fatty acids, right? I always think of like the meme of the avocado that says, but I'm the good kind of fat. So we do have some good fats that are not synthesized by the body, so they've got to be part of our diet, like omega-3s and omega-6, often found in different kinds of fish. And again, these are heart healthy, so these are the ones that we do want to consume and can also lower blood pressure as well. Next, we also have some waxes that come from lipids, right? These are hydrophobic and prevent water from sticking to the surface. You see these a lot on like feathers of aquatic birds and also surface of leaves from quite a few plant species, actually. And probably one of the more important things you'll hear about in this class in terms of lipids is cell membranes. So cell membranes are made up of something called a phospholipid. And this is a molecule that has two fatty acids as well as a modified phosphate group attached to a glycerol backbone. So the phosphate can be modified by addition of charged or polar chemical groups, and basically we have the phosphate and the glycerol up here contain the hydrophilic head. So this is our water loving part. And then we have our saturated fatty acids or unsaturated fatty acids down here that make up the hydrophobic tails, the part that tries to stay away from the water. So when these come together and create a phospholipid bilayer meaning there's two layers here, we have our tails sticking in towards each other, again, avoiding all of the water. And then we have our hydrophilic head groups that are facing both the outside and the inside of the cell, where they're going to have or be in contact with aqueous solution. So the cell is our basic structural unit of life, right? And all of them have kind of the same basic architecture. We've got essentially, we'll call it water or some aqueous solution inside and outside as well. And the thing that separates these two is usually our phospholipid bilayer. So again, hydrophobic in the middle, hydrophilic on the ends. And within our phospholipid bilayer, we do have lots of different proteins that are embedded within, and these essentially help do quite a few different things. Okay, so we're going to kind of shift gears as well. I want to be able to talk about water. Water is very important to life on Earth, as you well know. And one of the main properties of water that we really like is that it's what's known as a universal solvent. So all of life's chemical reactions take place in water. And a lot of things can't really survive even more than a few days without it, right? Um, Anyone know what a solvent is? Solvent versus, let's say, solute, right? So a solvent is basically just a substance in which other substances can dissolve. So like salt water, we put salt, which would be solutes, into water, and then we create salt water. Okay. Water transports dissolved molecules, which is our solutes, from a place to place in a solution. So in this case, solvent is our water, solutes is our salt, and we create a new solution, salt water. So why is water such a good solvent? Why can it dissolve most things? Well, the reason is because we have our awesome molecule H2O. And what happens is we have a more negatively charged oxygen side and then a slightly more positively charged hydrogen side. So what this means is that our electrons aren't really shared equally between the atoms within our covalent bonds. And so we get a partial negative charge on one end and a partial positive charge on the other end. So our water kind of has two different poles, similar to a magnet. And we refer to this as polar. So you'll hear about polar and nonpolar molecules, but water is polar. So again, our more negative end of water molecules surrounds, when we're talking about like our example of table salt here, we have our table salt that's ionically bound together. And what'll happen is the more negative end of the water molecule will surround the positive sodium ions. And the more positive end is actually going to be attracted to all of our negative chloride ions. So these kind of surround each other, and they pull these molecules or these atoms apart, and that's what actually dissolves them. That's why water is such a good solvent. Other thing that we love about water is that it's sticky. So I'm sure most of you don't think of water and think, oh, it's sticky. But think about what water is actually made of. We've got these H2O molecules here, right? And again, we've got a polar molecule, but the way it's held together is by these hydrogen bonds. And remember, hydrogen bonds are really weak, and it can form between two polar molecules. So you've got that partial charge on the hydrogen side, the negative charge on the oxygen side, and that allows the water molecules to actually stick together. So some good examples of this, we have both cohesion, and that's when the water molecules stick to one another. So cohesion, like together, these ones are sticking to one another and actually creating something called surface tension. So you can see here things like insects, or if we ever see water flowing up tree trunks against gravity, water surfaces that insects can land on, when raindrops fall onto other like puddles of water and things like that, the water kind of sticks together. So this is the water molecules sticking to one another and creating something like surface tension, where obviously you've got to be a little bit spread out to stick like we normally would, but that's what cohesion is. And on the other hand, we have adhesion. So this is when the water molecules stick to other polar molecules that are not water. And the best example of this, I think, is usually water pooling on leaves, like after it rains. You can also see water kind of sticking to animal tongues if you're looking that closely and if your animal is taking that slow of a drink of water. But this is adhesion. Hydrogen bonds also mean that the water stays liquid longer, so they can absorb a lot of energy before they get really hot and vaporize or turn into a gas. Which also means that our water molecules can release a lot of energy before they get cold and solidify and then turn into ice. So on Earth, as you probably know, most common form of water is in liquid form. And that's because it's really hard to make it ice or water vapor. The other thing that we love about ice is that it floats on water. So the density of solid water, which is ice, is actually less than the density of liquid water. And because of that, ice is going to float on the water. Why does that happen? How can the density be less? Well, now we're going to go ahead and take a look at our hydrogen bonds. So when our hydrogen bonds are on ice, they're really stable. They're not really breaking apart and they actually kind of spread out. On the other hand, when they're in liquid, our hydrogen bonds are constantly breaking and reforming with each other to try to find some stability in that liquid environment. So that's kind of a few of the properties of water that we really like and are really crucial to life on Earth. And now we're going to talk a little bit about pH, and then we'll be all done. But pH is basically measuring the amount of hydrogen ions you have in a solution. So you probably know that a neutral solution is at a 7 pH, which is essentially pure water. Okay. And what happens, why we call this pH and why we're measuring the amount of hydrogen ions in a solution is that our water molecules, our H2O, can actually split briefly into separate hydrogen, which is H plus, and hydroxide, which is OH negative ions. So in pure water, the number of hydrogen ions is exactly equal to the number of hydroxide ions. So that means that water is neutral, right? And we can kind of see it over here. We see the amount of hydrogen ions is equal to the amount of hydroxide. Okay. On the other hand, if we start getting into acidic territory, our pH is decreasing, right? What happens here is that the number of hydrogen ions is greater than the number of hydroxide ions. Okay. So say here anything less than a 7, which is again neutral water, that is considered acidic. And now if we go the opposite way, anything greater than 7, and we have more hydroxide ions and less of the hydrogen ions, then that's when we're considered a basic solution. pH homeostasis is very important in cells, so you'll hear this quite a bit as part of them regulating their own cells or our own bodies, is that we want to maintain a healthy pH and that the pH levels are pretty consistent. We can't really deal with much of a pH change in our bodies. And strong acids and bases are really highly reactive with other substances, which also makes them destructive to molecules in a cell. So we want to keep everything, kind of closer to the neutral area for the most part if we can. In addition, a lot of biochemical reactions can only take place at a certain pH. So kind of like our enzymes with a lock and key, all of these reactions have to take place within a certain pH as well. So a lot of organisms function best when their pH stays within a specific range. We do have extremophiles that can exist in really acidic or really basic environments, but for the most part, we're all kind of hovering somewhere around neutral pH, and we can't really go too far out of that range in order to maintain homeostasis. So this is when things like buffers come into play, right? We want to make sure that we're buffering our blood pH levels. And that's basically the way that we maintain internal solutions. So again, they've got to be pretty close to our neutral pH, but you can see the diagram here is showing the body's buffering of a blood pH level. So the blue arrows show the process of raising pH as more CO2 is made. And the purple arrows indicate the reverse process where you lower the pH as more bicarb is made. Okay. All right, I know this was a lot of information and you've done really well getting through it. You can always divide this up if you need to, but please take away the major concepts from these 2 chapters. Don't get too lost in all of the small details. Really just looking for you to understand what kind of the basic components of life are, what carbon-based molecules are, why they're important, what water is and why it's important, as well as what pH is. And we'll get even more into the macromolecules of life as we start moving forward in the course and once we get to nutrition as well. But for now, just know that we've got those 4 macromolecules of life and where you'll kind of see them as the semester progresses forward and how they're used in different parts of the body.